I actually have something that ties in to just before that, we're talking about GLP.
Okay.
If you're not going to be referencing GLP when a inspector comes in, what standards will they be following to ensure that the laboratory is fit for purpose?
Again, it's not the lab that we're inspecting. We're actually inspecting the study and how it was conducted, and the quality management systems, the items we've discussed before. Some of them were maybe described in some guidances. Perhaps in the future, the guidance would be more, elaborate more on these points. But we don't use GLP standards for when inspecting these studies. Does this answer your question, Mr. Lehman?
Well, I'm not sure. What standards are then being, when it talks about calibration of equipment? We don't have to do that now because we're not following GLP recommendations. Tracking of SOPs, we don't have to do that now. So what's the standard set of applicable methodology procedures associated to that study that inspector is going to look at to evaluate credibility of that study?
Sure. At least for the analytical part, I can say that we rely heavily on the bioanalytical method validation guidance that came out in 2018. In the near future, maybe next year or so, it'll probably be superseded by ICH M10. However, that's what we rely on mostly. Anything that we feel can impact data quality or data integrity is something would be of concern. And I believe Sam or Heron can perhaps address the other part, the other type of the study, the IVPT part, IVRT.
I'd be happy to, yeah. Heron, would you like to go first, or shall I comment?
Please go ahead, Sam.
Okay. So I think this is a really good question you raise, because in terms of, putting oneself in the shoes of, like, Dr. Powell, who's saying, what do I need to do to make sure that the quality management systems, that the infrastructure, that the SOPs, that the training, that all of these are compatible with the kinds of things that are going to support regulatory decision making? I think that's the key question. What's my responsibility to make sure that the studies are conducted using that infrastructure? If I'm not using GLP as a guide, then what am I using? Or equally, even if you tell me relevant principles of GLP, as you pointed out, Kendall, what exactly does that mean? I think one of the things that's coming out of this discussion is that we need, as a community, to be able to define what those specific elements are for IVRT and IVPT studies. And I think that was why it was so helpful, Kendall, for you to be able to provide a presentation that talked through how you interpreted it, how you built those things. And I think everything that you talked about there continues to be relevant. So the distinction is simply that we want to avoid confusion about the relevance of US codified GLP. This is the FDA GLP in particular, not the EPA GLP. But we want to make sure that there's not some confusion about the scope of FDA GLP. And that's part of the reason why we're beginning to make this distinction here, because IVRT and IVPT studies technically don't fall under the scope, the intent of what FDA GLP is really targeted at. However, part of the reason that the acyclovir cream guidance nonetheless talks about the relevant principles are because GLP has up to this point been that one reference that has all the components of a quality management infrastructure, components like making sure that your personnel are actually qualified to do these studies, that they're trained, that you have standard operating procedures, that good documentation practices are in place so that one can reconstruct the study. These elements are really critical and continue to be critical, whether or not you're following GLP or not following GLP. They just happen to be nicely organized in the GLPs. But FDA GLP also contains things for animal necropsy and animal feed and things like that that are clearly not relevant to IVRT and IVPT studies. Therefore, what are the relevant principles? Well, the relevant principles are some of what I've just talked about there. And I think those will continue to be relevant going forward. So kind of moving away from actually speaking about it as GLP is not a reflection that these elements don't need to be there anymore. It's simply a reflection and kind of an acknowledgement of the fact that FDA GLP really is technically for a different scope, but that these same considerations would apply to a quality management infrastructure that you would continue to want to have in place for IVRT and IVPT studies. And Sam, I don't know, even before Hiren jumps in, if there's any further comment you have on the, on my perspective here.
Yes, I agree.
Hiren, would you like to comment also?
Yeah, I totally agree with you. That I mean, what we are talking here. I mean, there's a great collaboration between the review discipline and the OSIS team within the FDA. When we assess this information, what we look as even what Kendall mentioned, that we look at the details because we are assessing the information from the reports, and our colleagues from the OSIS, they do expect the the actual side. So I defer that that regulation side to the OSIS team. But I mean, I can probably take one of the example that we were discussing yesterday. Sorry, I mean, Theo, I didn't mean to put you on the spot, but in the IVRT section yesterday, we were talking about the single point calibration versus the multi-point calibration, and you raised some interesting point. But it was like, I mean, we were like delay, and I don't want to touch base on that aspect yesterday. But since we have some time today, we can talk about that. You said that, yeah, I think there was some dilution issue, and you received deficiency. I don't know which one you are referring, but the expectation from the review team is, I think even if you go with the bioanalytical method validation guidance, if your study sample is outside the calibration curve, and if you are diluting that sample, the expectation, what is mentioned in the bioanalytical method validation is to have the dilution integrity quality control sample. So, I mean, we still follow, and as Kendall mentioned that, you're not gonna take all the principle from the bioanalytical method validation guidance, but you need to take certain aspect of that bioanalytical method validation, because these studies are there to support the bioequivalence of the taste product. This is not just the quality assessment. So this is, this is very important study, and we need to, assure that, there's an accurate quantitation of your analyte. So we as a review discipline, we do follow, as Sam mentioned, that the principle, that is my understanding when we refer in acyclovir cream, the principle of GLP, that's what I'm in an impression. So, but as Sam from OSIS mentioned that, or even Dr. Rane mentioned that, not all aspects fall into that category. So it's just as a community, it's up to us where we want to draw the line.